Hello, and welcome to our lecture on microbial genetics. In this particular lecture, we will be introducing the idea or concept of genetic evolution in prokaryotic organisms. We will then look at the structure of nucleotides and DNA molecules, and using our understanding of DNA structure, we'll then move into describing and defining DNA replication. So genetic evolution is a process by which genetic material in a species or in a strain of organisms will change over time. Bacteria are very rapidly evolving organisms, and they are one of our key organisms for studying evolutionary changes. And this is because of their rapid evolution. Think about it. An E. coli cell divides every 18 to 21 minutes. That means that we can actually see evolution in real time. And we will watch a video of some evolution in bacteria in just a minute. Bacteria also exchange genetic information. In order for an organism to survive over time, it must evolve, and that evolution is directly related to environmental factors. And organisms evolve in order to adapt to a changing environment. So we are going to take a look at how bacteria are evolving or changing towards a specific environment. And interestingly about prokaryotic organisms is they actually can evolve because of this very rapid evolutionary process. They will evolve into far beyond just the general species level that we are used to seeing in eukaryotic organisms. They can do all of these things, and their form of evolution or genetic diversity is through horizontal gene exchange, which is another lecture we'll be covering in this particular chapter. This is all in chapter 8. But horizontal gene exchange is a way for bacteria to exchange genetic material or obtain new new genes and new characteristics in order to adapt to an existing environment. So let's take a look at someone explaining a little bit of evolution.
The bug has this intrinsic or innate programming to try to survive, like all of us. That's what we do as living organisms. And the best way we learn how to survive is through adaptation. Right? So Darwin's, the thing that we all learned in high school, survival of the fittest, it's true, but it's really survival of the most adaptable. And that's really what resistance is about. This is purely a numbers game. And so when you do give a certain antibiotic and say you start missing your pills, so you're decreasing the dosing, then your killing is inefficient, right? And when you have inefficient killing, you have more rounds of replication. The bugs are allowed to persist longer. And the longer a bug persists, they multiply, right? So an average bacteria like E. coli, for example, or staph, will double every 20 minutes, right? So every time it doubles, and then it's 1 to 2, 2 to 4, 4 to 8, so on and so forth, right? There's chances of mutations, chances of events happening where the bug is allowed to adapt.
So over here on the left, I just want to show you real quick. This is a phylogenetic tree that shows the evolution of Nosocomial Mersa. We've all heard of Mersa, that methicillin-resistant Staph aureus. This is a bacteria that originated in a hospital setting. And it became the first cases of Mersa were actually discovered back in the 50s or 60s. But this phylogenetic tree, in order to show you how quickly, these are all different strains, different genetic strains of the same Mersa organism. And we're looking from 1982 here in Australia, 87, 96, all the way up here to 2007, when around 2010 is when this was created, but we're going all the way up to 2007 here. In this case, you're looking at from 1982 to 2007, you're looking at just over like 25 years. That's a really short period of time. I mean, that's just a blip on the radar in terms of evolution. So bacteria do evolve very quickly. So this is a video that is going to describe an experiment that was done just not that long ago, maybe about 5 or 6 years ago at Harvard. It's a very simple experiment, and it really shows evolution in real time.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So this experiment here really was a really fascinating way to show evolution in bacteria in this really rapid sense and how quickly it can occur. If you think about it, the normal antibiotic regimen for most patients would be a 10-day regimen. And all of that evolution and that resistance that developed in that strain of E. coli occurred over 11 days. So making sure that the antibiotics are at the right concentrations is really important, because if that concentration becomes weakened, then resistance can develop very easily in those organisms, as you just saw. Okay, so let's take a look now. Let's move into the molecular level of DNA itself. Since evolution is based on genetic exchange, we need to take a look at the DNA and the genetic material itself. And many evolutions are the result of mutations, and many mutations are the result of changes that occur during DNA replication, which is why we study DNA replication. But in order to understand DNA replication, we have to go all the way back to the molecular level and really kind of look at the structure of the nucleotides that make up the DNA, how DNA is built, so that when we move into replication, some of the directionality things that occur in replication will make more sense. So let's connect the dots here so we can get to the actual mechanism of replication. So here on the left are the DNA and RNA nucleotides. DNA and RNA are made up of 2 classes of molecules known as purines and pyrimidines. Purines are the adenine and guanine, and I use the analogy or the term pure as gold. So pure, purine, A for adenine, and G for guanine. So pure is gold. And then my pyrimidines are the everything else. The uracil, which is found in RNA only. Thymine, which is DNA only. And then cytosine, which is found in both. Adenine and guanine are also found in both DNA and RNA. So these are just the bases, right? These are simply the bases. And in organic chemistry and in biochemistry, when we have these carbon-based ring-like structures, or any kind of organic chemistry, we we give each carbon or each atom in the molecule a number. So here we have nitrogen 1, carbon 2, nitrogen 3, carbon 4, carbon 5, and carbon 6 in a pyrimidine. And then we have 9 locations, 9 structures in a purine. Purines have they're a double ring structure. attached, what these bases are attached to over here on the right, here's our base right here, and A is bound to a T. And what these guys are attached to are a sugar. And in DNA, they're attached to deoxyribose sugar. So this is called deoxyribonucleic acid. And in RNA, they're attached to a ribose sugar. And so that's RNA, which is just simply ribose nucleic acid. The difference between the two you can see is that this guy has simply an hydrogen and this guy has a full hydroxyl group, or OH. The reason this structuring is important is because I now have a ring structure, like you see over here on the right. I have a ring structure attached to another ring structure, which means that I need to number this one separately. And when I do that, I number the sugars with the word prime. So here I have carbon 1, so this is 1 prime. This is carbon 2, so it's 2 prime. And the prime is just written as a little apostrophe after the number. So here's 3 prime. Here's 4 prime, and then this carbon coming off of the 4 prime carbon is the 5 prime carbon. So all of the carbons are given a number. You can see here that the 1 prime carbon is what the nucleotides are actually, the bases are attached to. So the bases are going to take off this OH here, and this is going to become a base, whether it's a T, a C, or a G. Same thing happens here on the RNA. Right? So here's a base. Now, notice that DNA is a stack. The nucleotides are all stacked up on top of each other to create these big long chains. And the bases are what actually connect together to make the DNA double-stranded. So DNA is normally single-stranded, and then the bases are complementary to each other, and they come together and create this double-stranded or double helix that we're used to seeing. Now, in base pairing, this is called complementary base pairing. This occurs in DNA. A is going to bind to T. So adenine always binds to thymine, and it does so with 2 hydrogen bonds. Guanine will always bind or base pair with cytosine, and this will occur with 3 hydrogen bonds. When this occurs, the T to C, and then we see here A, T, G, C. This is the sequence of the nucleotides. So this sequence over here is going to be complementary to the sequence on the other strand. And since A always binds to T and C always binds to G, we can say that this is known as complementary base pairing. This was discovered and proven by a gentleman by the name of Erwin Chargaff. And there is something called Chargaff's rules, which is base pairing rules. And in Chargaff's rules, the percent of A plus the percent of G equals the percent of C plus the percent of G. I'm sorry, of C and T. Excuse me, that's a T. So if let's say 20% of the nucleotides found in a sample were A, then A always binds to T, so this means that 20% is going to be T. 20 plus 20 is 40, so that leaves 60%, and that means that 30% is going to be G, and 30% will be C. 20 plus 30 is 50, 20 plus 30 is 50, so I equal out. So this is known as Chargaff's rules, and these are base pairing rules, what we call complementary base pairing rules. Now let's come back over here to the left on our deoxyribose sugar here. It is important to know the numbering of these because when we build DNA, you can see here we have our nucleotides are attached on top of nucleotides. If you notice, this is occurring with a phosphate group here. And attached to these sugars are going to be 3 phosphate groups. This is why ATP is considered a nucleotide. When these nucleotides come together and they stack on top of each other, they bind at that phosphate. And 2 of the phosphates get broken off. These are called a pyrophosphate. They produce energy. The energy released by the breaking off of the two outer phosphate groups is what provides the energy for these 2 nucleotides, these 2 deoxyribose sugars, to stack on top of each other and form what's called a phosphodiester bond. But this is occurring here at the 5' carbon. The next nucleotide, which leaves this 3' carbon free or open. So the next nucleotide that comes in, its deoxyribose sugar will create that phosphodiester bond at its 5' sugar, leaving its 3' available here. This will continue in this same sequence over and over and over again for each of these nucleotides. So that if I were to flatten out my DNA molecule on the left here, what my carbon that is free is the 3' carbon. At the top of my molecule, the carbon that is free or not bound to anything is my 5' carbon. Now, in order for my nucleotides to face each other and be able to carry out complementary base pairing, my other strand has to be parallel to the original strand, right? So the one on the right is parallel to the one on the left. However, it is running in the opposite direction. So it's what we call anti-parallel. And it is anti-parallel because of this directionality. So that means that here, if you notice, basically my deoxyribose sugar here is flipped upside down. So this is my 3' sugar at this end. And of course, what do I have free here? I have my 5' sugar. So my strand on the left is running from 5' to 3', and my strand on the right is running from 3' to 5'. So that brings us to DNA itself. Here we're showing, this is just another diagram showing kind of the same thing. We have our nucleotides here. Here are the phosphate groups. You can see this is that 5' carbon right here, and there's the 3' there. So this is the 3' end because the 3' is not bound to anything. Same thing here, it just still has its OH, so it's 3' at this end. And then of course 5' is free right there, and the 5' is free right here, so it's the 5'. Here they're showing you where these nucleotides are together. You can see again G to C, A to T, A to T, A to T with 2 hydrogen bonds, and G to C with 3 hydrogen bonds. Now in bacteria and prokaryotes, the DNA in bacteria is located in 2 different places. We have the chromosomal DNA, so that's what basically encodes for majority of the proteins, everything a cell does metabolically, structurally. The chromosomal DNA is kind of the everyday workhorse genetic material. Plasmids are the secondary form of DNA, and plasmids are in addition to chromosomal DNA, and they bring unique characteristics. They add genetic diversity to microorganisms. DNA will always be replicated prior to binary fission, and that's what we're going to take a look at now. So genetics is simply the science of heredity, how things get moved genetically, how DNA gets passed on from parent to offspring, or in the case of bacteria, from 1 cell to 2 cells during replication. DNA is composed of, DNA itself is the genetic material. It's made up of the nucleotides like we just saw. Chromosomes are large sequences or large strands of DNA that contain genes. Genes themselves are the segments on DNA that actually encode for something that will go through transcription and translation and result in some type of structural or non-structural component. Plasmids, again, are those small circular pieces of DNA that are only found in prokaryotes and carry genes that are in addition to chromosomal genes. The genotype of an organism is its genetic makeup, and the phenotype of an organism is the genetic expression of that genotype. So the gene is the DNA sequence that results in a protein. The protein is the actual phenotype. Now, prokaryotes have a unique property known as operons. Operons are where genes of similar function, or genes that are related in their function, meaning they're used in a very particular pathway or in a very specific type of metabolism. Those genes are grouped together under a single regulatory region. And this is because bacteria are incredibly small. It's a space-saving measure. And they take these genes that would only be expressed together in specific instances or in a specific environment, they group them together and they put one regulatory region in front of it. And this way they can regulate those genes on a single level as opposed to a multiple level. And this drastically reduces the size of the chromosome and the enzymes necessary to regulate and either promote or inhibit expression of those genes. So those are referred to as operons. And operons are they are unique to prokaryotes. We will study the lac operon in I think lecture 4. So DNA replication as a process is what we refer to as semi-conservative. This is because each of those strands that create that DNA structural molecule, those strands will separate and become parent strands to the daughter strands. And the resulting cells from binary fission will have one DNA strand from the original cell and one new synthesized strand that was created during replication. So it's conservative in that we maintain one of the strands, but it's semi-conservative because each cell only receives one parent strand. In order for DNA replication to occur, it requires a set of enzymes. It requires DNA polymerase 3. This is the enzyme that's responsible for actual synthesis of the new DNA strand or what we call daughter strand in replication. DNA polymerase 1 is a polymerase that is going to replace a primer. We'll talk about primers. Helicase is an enzyme used to unwind DNA and create the replication fork that we'll be looking at. Topoisomerases are classes of enzymes that prevent over-twisting of DNA. Think about a landline phone cord. If you try to untangle a landline phone cord by pulling the strands apart, the helix that occurs where you pull will increase in torque, and this will eventually cause it to break. So DNA gyrase is the enzyme found in bacteria that reduces torque during replication in order to prevent the DNA from over-twisting. There is a class of proteins called single-stranded binding proteins, and these guys are going to protect single-stranded DNA during the replication process until it can be complementary base paired. DNA primase is what lays down the primer that polymerase 1 is going to later remove, and that primase lays down a primer in RNA nucleotides instead of DNA. And lastly, we have DNA ligase. DNA ligase is a enzyme that's going to join together fragments that have been replicated and repair the sugar phosphate backbone. Now, DNA replication occurs in two forms. Both of these will be occurring at the same time. One strand will be replicated as a leading strand. The second strand will be replicated as a lagging strand. So in bacterial replication of plasmids, remember plasmids are those small circular pieces of DNA that are in addition to chromosomes. When plasmids replicate, they have a single origin of replication. That origin of replication is where all of our enzymes responsible for initiating and following through with DNA replication will kind of join together. It's a very specific DNA sequence that will be recognized by helicase and DNA gyrase. They will create what's called the replication fork. And the replication fork is where the two single strands of DNA are where the double-stranded DNA is separating into two single strands. Now here, the dark purple strands are the original strands, and the light purple strands are the ones that are being replicated as our plasmid is being pulled apart, as it's being separated. Notice that this replication is what we call bidirectional. So replication is occurring at the left and to the right at the same time. This is going to cause these plasmids to eventually peel away from each other until they reach a sequence known as the termination sequence. At the termination sequence, replication is completed, the complex falls apart, and we now have two plasmids. Okay, so here we have a diagram that is showing you the overall process of DNA replication. And DNA replication is really kind of a 6-step process. Now, first we have our parental DNA here, and we need to create our replication fork, because in order for complementary base pairing to occur, we have to make the DNA single-stranded in order to replicate it. So DNA helicase, that's this green guy right here. So here's helicase. Helicase and topoisomerase, or DNA gyrase, both occur together as a complex, and they will create our replication fork. Once the DNA becomes single-stranded, we now have these bases that are kind of free hanging out there, and DNA bases want to be bound to another base. So in order to keep the DNA from zipping back up together, preventing synthesis of a new strand from occurring, or from to keep these bases, these single-stranded bases on the DNA molecule, on the parent strands, from just kind of randomly binding nucleotides that are floating around in the area. Single-stranded binding proteins are added, and these single-stranded binding proteins protect the single strands of DNA. They will be removed by DNA polymerase during synthesis. Now that we have our replication fork created and we're protecting our single-stranded bases with single-stranded binding proteins, we're ready to begin the replication process. And the first thing we have to do is lay down a primer. The primer is just simply a way to get things started. So primers in DNA replication are very temporary. They're going to later be removed and replaced with correct DNA nucleotides. So in order to make sure they're easily recognizable, primase, the enzyme that lays down the primer, uses RNA. So RNA nucleotides, it's referred to as an RNA primer, because the nucleotides used to create this primer are RNA nucleotides. This makes them very easily recognizable by correction mechanisms, and they will go in and cut out that primer and replace it with DNA nucleotides. But the primers are there for a very distinct reason. And the reason for that is because we have directionality in DNA. And so this is why we covered the structure, the nucleotide and DNA molecule structure, a few slides earlier. In order for DNA polymerase III to be able to synthesize a new strand, it has to have an existing 3' carbon available to it for the incoming 5' carbon of the incoming nucleotide. So the reason that primase lays down this RNA primer is because we need to have one of the carbons on the sugar phosphate, in that sugar phosphate backbone, we need to have that free. So since this is the 5' to 3' strand up there, what we're going to have here is we're going to have a 3' carbon available for the incoming 5'. So this is the 3' carbon right here on the end of this primer. And when DNA polymerase III comes in, DNA polymerase III has to have an existing 3' carbon available to covalently attach that incoming 5' carbon. So that when we get all the way to the end of it, right, we have a 3' carbon here. Because remember, we have 5' to 3', and the complement strand is 3' to 5'. So if I want to bring in a new nucleotide in, as a DNA polymerase molecule, I'm going to bind the 5' carbon to this one. And that means that I need to have a 3' carbon already available. So the primer is laid down in order to provide a 3' carbon to get DNA replication started. Once this process starts, then every time a new nucleotide is laid down, this is the 3' and that's the 5'. So it's just constantly, because of the structure of these guys, they're constantly providing, the new DNA nucleotides are constantly providing those 3' carbons for this replication to occur. Now the problem we have over here at the bottom strand, what we call the lagging strand, this up here is the leading, and leading strand synthesis is going to move towards the replication fork, and it is what we call continuous, because once DNA polymerase III begins replication, it just follows along the replication fork. But in our bottom strand here, this is referred to as the lagging strand. And the lagging strand synthesis is discontinuous. It is discontinuous because we must do this in small pieces called Okazaki fragments. And directionality is the name of the game. So since my top strand here is running from left to right, 5' to 3', and it's able to follow the replication fork in synthesis through DNA polymerase III. But the problem I have with my lagging strand here is it runs 3' to 5'. So if I lay down a complementary base, when I lay down that complementary base, my 3' carbon is actually over here, right? So I'm just gonna, there's the primer, right? Here's my primase laying down a primer. That's what's in red. My RNA is in red here. And that leaves my 3' carbon on the opposite side of the replication fork. So this means that my replication is going to be occurring in the opposite direction of the replication fork. If I lay my primer down here and I just was continuous, then from left to right, then all of this DNA over here would not get replicated. Instead, what the cell does is the bacteria pull this DNA out in loops and then they replicate it backwards. And they do this in small pieces. Now you'll notice over here is another primer. And remember, primers are made out of RNA nucleotides. So when DNA polymerase 3 here, right, here's that 3' carbon that the primer is making available, binding to the 5' carbon of the incoming nucleotide. As DNA polymerase moves to the right, it's going to bump into the primer, right? It's going to bump into the primer from the previous strand that was replicated. When it does, it's going to fall off. And I will have this fragment, right, in blue here, light blue here is my fragment of DNA that has been replicated. Now, Okazaki fragments are not particularly long. They're not huge. And so what we have here is this same type of replication. It's just occurring repetitively. Now, once my polymerase 3 has fallen off, I still need to come back and I still have to replace this primer here. So DNA polymerase 1 is shown here, replacing the primer with DNA nucleotides, RPLA. Okay, so we're going to replace the primer. Sorry, my writing is pretty bad here. Replace the primer with DNA nucleotides. And this polymerase 1 is the enzyme responsible for replacing that primer. But one thing polymerase 1 cannot do is polymerase 1 can't create that sugar phosphate backbone. It leaves a dent or a little nick in that sugar phosphate backbone. So our final step is to then proofread the DNA molecule with DNA ligase. DNA ligase fixes that break in the sugar phosphate backbone so that our new strand is now complete. So I'm going to, on the next slide, I left this blank on purpose because I wanted to draw it with you. One of the best ways to see DNA replication, aside from maybe watching a video, is actually to draw out the steps and write them in order. So in step 1, we have DNA helicase. I hope my pen works with us here. DNA helicase and gyrase. I'll just make it one word here. We create the replication fork. So that's step 1. We got to create that replication fork. So I'm just going to draw my DNA down here. And my complex here. So this is my helicase. This is I'm just going to write a G here for gyrase. And I'm creating my replication fork. In step 2, let's make this 5' and this end is 3'. So this is 3' and this is 5'. In step 2, my single-stranded binding proteins are going to bind. And they're binding to the single-stranded DNA. So if I have all of these nucleotides here, just put them out here like that. Then they are like little caps, little plastic caps that are going to sit on my single-stranded DNA so that I don't get any innocuous binding, right? They're protecting it. Then I'm not going to draw that on the rest of them because I want to be able to use that template. And now in step 3, I'm ready to start replicating. So my primase is going to lay down the primer, which remember is in RNA molecules. Let's see if we can't change colors here just to make things a little easier to see. So we'll make our primers red like the last picture. So I'm just going to lay down my primer here. Excuse me. And remember, my primer is in the opposite direction. So if this is the 5' carbon here, this is the 3' carbon of that last nucleotide, that last RNA nucleotide in my primer. So now I'm at step 3. I'm going to come back to my pen here. I'm going to change colors again. And I'm going to move into step 4. In step 4, DNA polymerase 3 is going to begin synthesizing. So it's going to synthesize the new DNA strand. And we'll make that green. So now my incoming, right, so here I have my polymerase 3. Remember, if this is 3', that means that this carbon, this is the free 5'. So that's where this is going to be joined together. And polymerase 3, every time it lays down a nucleotide, that leaves a new 3' there, so the incoming nucleotide's 5' can attach. And polymerase 3 is going to move towards the replication fork. This is continuous. My writing is terrible, I'm sorry, but my computer is not agreeing with my pen here. So I have continuous replication here. It's moving towards the replication fork. This is my leading strand synthesis. Now, I do need to move into step 5 and 6, however, because I still have this RNA primer laid down, and I need to do something with this RNA primer. So I'm going to come in with polymerase 1. So in step 5, we'll just leave this color here. We have DNA polymerase 1 replaces the primer. It replaces the primer and is going to kind of create this. It's going to replace those RNA nucleotides with DNA nucleotides. So it's polymerase 1. Polymerase 1 will replace the RNA nucleotides of the primer with DNA nucleotides. And then we get into step 6 here. And in step 6, we'll go ahead and make this a new color as well, just so it's a little easier to see. And we'll go with magenta. So here we're going to then use DNA ligase to correct or to repair the sugar phosphate backbone. So this is step 6. We'll just write ligase here. And DNA ligase will repair that sugar phosphate backbone that was created there. Now remember, down here in the bottom strand, in lagging strand synthesis, we are doing the exact same thing. It's just going to be occurring in the opposite direction. That's the only difference. In this case, our lagging strand is moving into the right or away from the replication fork. But I go through the same 6 steps, just in the opposite direction. So in step 1, DNA helicase and gyrase are creating that replication fork, which has already occurred. In step 2, my single-stranded binding proteins, here they are, even on the lagging strand. They're protecting my single-stranded DNA. Then I'm going to move into my next step. I still have to lay down a primer. So I lay down my primer. And in this case, we'll lay down the first primer is laid down over here. Here's my primer. This is going to be the, this is the 3' carbon here. This is the 5' carbon here. DNA polymerase now is going to come in. And DNA polymerase is going to replicate, but it's looking for that 3' carbon, right? Because it has to attach that 5'. So this is my polymerase 3. So this is my DNA polymerase 3. Now I have all of this empty space right around here, so I need to lay down new primers. So what's going to keep happening is these primers are just going to keep getting laid down, and we're going to go through DNA replication. We're going to go through leading strand synthesis over and over again. I'm trying to get my pen color here to work. It's not working, so we'll just do new primers are here. This is DNA that's coming over this way. This is polymerase again 3. And we're creating these Okazaki fragments. So there's step 3, and then step 4, DNA polymerase 3 is creating them. But when polymerase 3 runs into this primer right here, it's going to fall apart. And I then move into step 5, where DNA polymerase 1 comes in and replaces the primer. And then in step 6, DNA ligase comes in and just repairs that sugar phosphate backbone. So this is going to be occurring in small fragments. And again, these are called Okazaki fragments. It works a little better if I write a little slower. So they're referred to as Okazaki fragments. So in review, in order to review, just ask yourself some of these questions, and these are the things that you need to be able to understand and apply. What are the different enzymes that are involved in DNA replication? What is the function of each of those? What do they do? How do the functions of DNA polymerase 1 and 3 differ, and how are they the same? So what do they do that's similar, and maybe within their mechanism, do they work in very similar ways? What's meant by semi-conservative? What's an Okazaki fragment? Why is DNA replication continuous on leading strand but discontinuous on the lagging? You should know all 6 steps of DNA replication, be able to draw those steps, and be able to label a diagram that shows you, be able to label a diagram where different enzymes are located or what is happening at those different locations in a replication fork. You should also be able to apply some basic principles of genetic evolution and see how DNA replication relates to evolutionary changes in bacteria. So that's it for lecture 1. We will move on to transcription and translation in lecture 2.